involved in auditory and visual attention. Prior to joining Northwestern, Dr. Braga trained with Rodney Bruckner at Harvard and as a Sir Henry Wellcome postdoctoral fellow, where he developed an individual-focused approach to studying distributed networks with fMRI. Dr. Braga then trained in intracranial electrophysiology with Joseph Parvizi and Russell Poldrack at Stanford as a K99 Pathway to Independence Fellow. Please join me in welcoming Dr. Braga, both for the AD seminar and to our department. We're very pleased to have you.
Thank you so much, Josh and Lisa. Thank you for the invitation. It's a real pleasure to, thank you for the virtual hand claps. It's a real pleasure to be here to share some of my work with you. I've just started at Northwestern a month ago, and I really look forward to meeting everybody and discussing potential projects and hearing about how the department's doing. So with that, I'm going to share my slides. Can you all see these slides?
Yes.
Yeah, okay, great. So yeah, as Josh explained, I'm interested in association cortex, and just to give you some background as to why one might be interested in studying these regions in particular, the human brain has expanded dramatically over our recent evolution, and you can see that just in terms of cranial capacity, but also in terms of the surface area of the cortical sheet itself. However, this expansion hasn't been uniform. If you take the regions of a monkey's brain, so cytoarchitectonically defined regions in a monkey's brain, and you'd look at the homologous regions in a human brain as best as you can calculate how much you have to grow the monkey brain region to reach the human size. And you can make this map of how much the human brain has grown in a comparative way. And if you look at this map, you can see the cool colors here are regions that have not grown as much, have not expanded as much as the hot colors here. And the cool colors in blue broadly just overlap with primary sensory regions. You have the somatomotor strip here and the visual cortex at the back. Those regions actually haven't expanded that much. Whereas if you look at the regions in between, these are the association cortices, they have expanded dramatically, something like 20 to 30 times. Another way to show this is if you just, here we take the brains of several of our evolutionary cousins, and we color in the primary sensory regions in colors here. By doing that, we can estimate what the brain of our common ancestor looked like. And that brain, probably most of it was devoted to primary sensory processing. Whereas if you look up in the humans here in the top right, actually the primary sensory regions take up a small percentage of the brain, and most of the brain is taken up with the in-between stuff, so the association regions. So if me as a cognitive neuroscientist, I care about things that humans are good at. For example, language is a great example of that. But if we care about neuropsychology, looking within association cortex and understanding the organization of association cortex is a good place to start understanding the brain cognition relationship. So just to go over a brief overview of my talk, I'm first going to look at the organization of association cortex. I'm going to show you how association cortex is organized into parallel distributed networks. I'm then going to talk about some of the limitations of group averaging in fMRI. And then from there, I'm going to talk about the observation of parallel distributed association networks in individuals using fMRI, and talk about evidence for functional specialization of these parallel networks. I'm then going to go on to show you evidence that the language network is a typical association network when you consider its topography in relation to other association networks. And finally, I'm going to present evidence that local functional regions might influence the specialization of these distributed association networks. So that's a lot of words, and hopefully as I go through the talk, one by one, these things will be clearer. So let's start by looking at the organization of association cortex. And for that, I'm going to present this. This is a work by Patricia Goldner-Rikic in the late 80s. And this was a study where they did a dual labeling experiment within the same monkey. So they injected radioactive traces in the prefrontal cortex and the parietal cortex of these same monkeys. These are both anterograde labels. And what they did is, these injection sites are really far away, but they were looking for regions in the brain where we have convergent projections from these two different injection sites. So here I'm going to show you two adjacent slices which are taken from the anterior cingulate cortex. And these slices are side by side. One is stained to look at one tracer from the prefrontal cortex, and one is processed to look at the other tracer from the parietal cortex. The arrows are meant to show you anatomical landmarks so that you can compare across the two slices. And what this is trying to show is that the prefrontal cortex injections project to this region of anterior cingulate cortex that's shown here, the bright column here. Now the parietal cortex injection site, which again is really very far away in terms of cortical distance, also projects to the same patch of the anterior cingulate cortex. So these two distant regions are having convergent projections to this part of the anterior cingulate cortex. If we then look at another part of the brain, this is the parietal area 7M, so more posterior medial cortices. Again, the prefrontal injection site is projecting to this medial region, and the parietal injection site is also projecting to regions that are very close by. So again, both of these association regions are having convergent projections to the same sites elsewhere in the brain. And they observed this feature in multiple brain regions and came up with this idea that association cortex is organized into these into these distributed networks that interconnect many different association cortices. And so this is illustrated by this diagram here, where the regions of association cortex are projecting to multiple different other regions of association cortex. And another cool thing that they observed was that regions that sit side by side within one part of the brain, so in this case, two regions that are side by side in parietal cortex, seem to project to regions that are also side by side in the prefrontal cortex. So you have this interesting parallel behavior where, as I've described, two regions that are projecting side by side to each other. And so from these few observations, Patricia Goldman-Rukic surmised that it's not just one broad network. Actually, it seems like association cortex may be organized into these parallel networks that they share the same broad spatial motif, or the shape of the network, the distribution of regions, but they have this parallel organization where there are regions that are side by side in one part of the brain project to regions that are side by side in another part of the brain. And this is very important to what I'm going to talk about later. We are seeing this kind of pattern in humans using fMRI. So I study fMRI, and we can actually already see glimpses of this. We've known about glimpses of this these same patterns for many years now. So here is a seed-based correlation map where Thomas Year and colleagues placed this seed in the parietal lobe, and in the colors you can see, in the red and yellow color scale, you can see the correlation map from this hand-picked seed. So you're seeing correlations in the lateral temporal cortex and the dorsal and ventral prefrontal cortex. And that already resembles what we're seeing in terms of what Patricia Goldman-Rukic described. If we looked at the lateral surface here, parietal lobe, superior temporal sulcus, and PFC, we're basically seeing a similar pattern, bearing in mind that on the left is a human brain and on the right is a macaque brain, and they expand. The regions are in slightly different places. And also, if I showed you the midline, you would see a similar distribution of regions, of posterior and anterior cingulate regions, and the parahippocampal gyrus for this network. Now, this map that I showed you on the left is made by a hand-selected seed-based functional connectivity approach. We can also do a parcellation approach where it's agnostic to any particular seed. It just clusters all the correlations in this brain into clusters or networks. And using these approaches, we've been able to identify different large-scale networks. For example, the default network shown in red here, the frontoparietal control network shown in yellow, or the dorsal attention network shown in green. And so this has been a tremendous engine for discovery. We've been able to map these brain networks within individuals using fMRI, and I think that's great. But these maps are made using a group averaging approach, and I'm now going to discuss some of the limitations of that using this example here. Now, if I showed you this image and said, what is this an image of? You'd probably have no problem telling me that this is an image of a face. However, if I asked you, who is this an image of? You probably would struggle to tell me that this is the face of Natalie Portman. The way this image is made is they took all the stills from the film The Black Swan, which had faces in them. They aligned those faces, and then they averaged across all of those faces. And so what happens when you do this group averaging is features that are shared across faces, so the location of the eyes, the location of the nose, the mouth, the broad shape of the face, those things are shared across individuals, so those things get accentuated by the group averaging. However, we miss out on important idiosyncratic details that are important for us to identify this person. So details of Natalie Portman's face are just lost by this group averaging process. And we have a similar problem with fMRI. So the map that I showed you, for instance, was made by scanning 1,000 different individuals, just a few minutes of scanning within each individual. And then you align the brains as best as you can, and then you compute an average across all of those brains. And so the image I just showed you is likely to be just like the Natalie Portman's face I showed you a second ago. It's a group average. It's a central tendency, but there are important details that are likely to be missing. In 2015, this very important paper came out by Timothy Laumann and colleagues from WashU. And in this paper, Russ Poldrack went into the scanner a few times a week for about 1.5 years and scanned himself many, many times. And by doing so, he had a lot of data from his own brain. So he could just do the same averaging approach, but just within his own brain. And by the way, I forgot to say, but the reason we need to do this averaging is because fMRI is typically a very low signal to noise ratio technique. And so this was a way for us to get over that noise barrier by just averaging across individuals. So it was very useful to do the group averaging. But what Russ's approach did was he was able to just average within his own brain and therefore preserve his own idiosyncratic anatomy and make maps of his own networks. He wasn't the first to do this. There are many others who've done this, notably Nancy Canwisher, but in vision, this was something that's more widely done than in cognitive fMRI. But so just to zoom in on those brains, here is that group average on image on the left, and here is a seed-based correlation map from this seed here. And then here is the same location and pretty much showing the same network, but in Russ's brain, you can see there are details here. The group map looks like a blurry version of this map based on Russ's brain, right? And so when we saw this, we hypothesized that maybe there are important details here that if we have enough individuals and we make the maps from enough individuals, we can see new details. It's like taking a leap forward in spatial resolution. Maybe there's something that we can glean about the brain by doing this. So I just told you that association cortex is organized into parallel distributed networks and showed you some of the limitations of group averaging in fMRI. So what we did is we decided to replicate Russ's approach, but this time we did 4 individuals so that we could look for patterns that were consistent across 4 individuals. Each individual underwent 24 fMRI sessions, and this was work I did with Randy Buckner at Harvard. Here was a typical setup, typical protocol for one individual. First, they would come in for an orientation. We'd explain the study. Then they would do some behavioral tests, and then we would have a week where we would do 3 different scanning sessions within that week. This allowed us to get a feel for whether that participant was going to be able to tolerate the scanning protocol, which was quite intensive and extensive. And it also allowed the participant to see whether they really wanted to be involved, knowing, some people just are not comfortable in the scanner. So that was a very useful thing to have. And then at the end of that week, we would have a debrief. They'd get a milestone payment, and we discuss whether to carry on. And if they continued, we basically scanned them a couple of times a week. And ended after 16 different weeks with a break in the middle. We had milestone payments to keep them engaged throughout the experiment. And we also did hearing tests because of the density of scanning. We wanted to be sure that we weren't damaging their ears. And we didn't see any effects of the repeated scanning on their hearing. This is a typical MRI session. So one of those yellow bars that I was just showing you. In this protocol, we actually did lots of different tasks, these micro tasks that lasted 2 minutes, because we wanted to do functional mapping of different domains. But actually the data I'm going to show you is from this 7-minute resting state scan. And since this study, I've been collecting data that's more I've been collecting more of the resting state data in the same session, so you don't actually need 24 sessions to do the kind of analyses that I'm going to talk about. We also benefited from, at the time, recently emerged technologies from the Human Connectome project, like high-strength gradients, 64-channel head coils, and simultaneous multi-slice sequences that were put into Prisma. And we have the same technology here at Northwestern, which is great. In terms of pre-processing the data, I'm not going to go into it in too much detail, but basically we had to rethink how we were pre-processing, because the whole point of the study was to optimize, to preserve the anatomy of that one individual. So we devised our procedures to optimize the alignment of that person's data to itself. So data from different runs and different MRI sessions had to be aligned in order to do these analyses. So the pipeline, which we called IPROC, was all about optimizing that. We also combined all the different registration steps into a single interpolation, because that reduces blurring, and we wanted to be able to tease things apart. We also ended up upsampling our functional MRI data to 1 millimeter space, which creates a really huge files, really hard to handle. But that allowed us, I think, to preserve some of the resolution and be able to tease things apart even further. And finally, at the very end, we did a very minimal smoothing of 2 millimeters, and 4, 6, 8 millimeters is common in fMRI. So we tried to really keep that down as much as possible. And here is just an example of the data quality. These are temporal signal to noise ratio maps. These vary for a lot of reasons, but I just wanted to show that we have pretty even coverage throughout the brain, including in these very problematic regions like the ventromedial prefrontal cortex, where, I don't know if you can see my mouse, but I'm pointing to it. Here, usually you have a lot of dropout here. We actually got good coverage of that. You can see some of the dropout in this second patient, but generally we still have coverage there, and just generally high SNR throughout the brain, so the data was good. And our general approach was we had 24 sessions. We divided this into 2 data sets. One, a discovery data set of 12 sessions, where I freely explored this data set, and I handpicked seeds and looked at the correlation maps and tried to see interesting patterns in the data. Once we had patterns we were interested in, we then turned to an independently collected hypothesis testing data set that included the other 12 runs. So that was for statistical tests. And then finally, we combined all of the data into one so that we could get the best estimate of the network patterns that we were seeing. And so I'm now going to talk to you about some of the findings we made. So here is a very famous group average parcellation of the brain into 7 networks. It's very widely used. And I'm just going to focus on the group-defined default network. I'm going to take out the red color and just leave the boundaries. And now I'm going to show you 2 seed-based correlation maps from subject 4, from one of the subjects in our study. So here is the correlation map from this one seed. And what you can hopefully see is that the high correlation regions are, generally speaking, within the bounds of the default network. So we think we have the default network here. Now, if I show you the map from the other seed, this seed also produces a correlated network that is within the bounds of the default network. But importantly, they occupy distinct regions. So I'm going to flick back and forth between those, so you can see that they're actually occupying distinct parts of the cortex. So these 2 networks are separable, but both of them are within the default network. So I'm just going to leave both of those up, one in red and one in pink. We just call these network A and network B, default network A and B, for simplicity. But what you can hopefully see here is both of these networks are within the bounds of the default network, the group-defined default network. And if I asked you to pick which one is the default network, you maybe would struggle to do so. Now I'm going to take away the boundaries of the group network, and I just want to focus on a couple of things. So if you look at the lateral temporal cortex here, you can see just how closely packed these networks are. The regions of network A are almost surrounded by the regions of network B here. And if we now focus on the medial prefrontal cortex, the regions of each network are interdigitated in a really complex relationship. You can see why even a small amount of blurring might just completely lose this organization, because the 2 networks will be blurred together. Perhaps the most important thing that we observed was, if I'm now highlighting 9 zones of the cortex, and in each of these zones, you can see regions from both networks sitting side by side, right? Even in some of these smaller regions, like number 6 here in the middle singular cortex. So why this is important is because it suggests, it provides evidence for what Patricia Goldman Rakic was arguing for, that these two networks are sitting side by side and they have the same distributed shape. So it's evidence for these parallel networks observed in this individual. Now, this was in one individual. We also saw this in all 4 individuals in the original study. Here is a different one, subject 1. We've also now replicated this in, I think around 20 to 30 individuals now. And I've even seen this at Stanford in a different scanner altogether. Yeah, and so we were very excited by this. We also looked at other networks like the frontal parietal and the dorsal attention networks. And in these, we also saw evidence of separable networks that had the same parallel structure, which was exciting. One thing that was particularly cool that I just wanted to share was these networks seem to form progressions that repeated in different parts of the brain. So for instance, here I'm going to zoom in on the lateral temporal cortex and the medial prefrontal cortex. And I'm going to show you the networks one by one. So these are the 2 default networks. Here are 2 frontal parietal networks. And here are 2 that we observed in the dorsal attention network. What you can hopefully see is that there is a progression here from the reds to the blues to the greens, which is replicated here in the medial prefrontal cortex. So reds to blues to greens. And in other parts of the brain, you also see this similar relationship. Why that's interesting is because, again, it suggests that association cortex, a principle of association cortex is that it's organized into these parallel distributed systems. And this is something that's been remarked before. For example, this paper by Daniel Margulies, talked about how these sequences are found in multiple parts of the brain and suggesting this parallel organization. Okay, so I've just talked about how we can observe these parallel distributed association networks within individuals using fMRI. And one thing you may be asking yourself is, how does this organization emerge? And one idea is that early in development, association cortices are wired into this broad distributed motif of projections. Large parts of the association cortex may already have this organization when regions start to specialize. And so by some process, maybe by an activity-dependent process, parts of the association cortex specialize, and that specialization occurs at the level of the entire network. So it's not individual regions that break away. When a region specializes, it inherits this distributed shape from just the wiring of the association cortex. So that's just one hypothesis. And if that were the case, these networks should also be functionally specialized as an entire network rather than individual regions of that network being specialized independently. And so I'm now going to show you evidence that we can see that within humans, individuals. So here are the 2 networks that I presented before, network A and B. 2 very interesting differences between them were that network A had this connection to the medial temporal lobe, the posterior medial temporal lobe, that network B did not have. We didn't see evidence of that from network B reliably. Whereas in the parietal lobe, network B was more anterior to more in the TPJ, temporal parietal junction, than this network B, which is always more posterior. And so if we look at group average data, people have made this comment for a while that multiple different tasks will activate the default network, but there are subtle differences between some of those tasks. So autobiographical memory tasks activate the map that is derived from an autobiographical memory paradigm. You typically will include this posterior medial temporal lobe, whereas other tasks that probe theory of mind, and this is when you're asked to think about what somebody else is thinking, that's termed theory of mind, they typically will activate this TPJ region, which is more anterior to an autobiographical memory task. And so we decided to test these 2 tasks within individuals and see if we could dissociate these networks functionally. And just to emphasize that, I also looked at these networks at 7 Tesla. Here is a 3 Tesla map and compared to a 7 Tesla map. Within the 7T data, you can actually see that the networks follow the gray matter ribbon, which is really nice. And with that extra resolution, we even saw that for default network A, not only did we see a posterior parahippocampal cortex region, but we also saw an additional region within the subiculum. So it's likely the default network A is very much hippocampal associated. But returning back to the task, so this is where we did with Lauren DiNicola, who's a graduate student in Randy's lab at Harvard. We had the same individuals come in 4 times, and we collected resonance state data to do network mapping, but we also had them do theory of mind task, which I'm going to go through in a second, and an episodic projection task where they had to remember the past or think of the future. And just briefly go over an example, the episodic projection task, participants were asked things like to recall a specific event that you attended with friends while in school. Was the event a sporting event, performance, or something else? They had to just think about it for a while and press 1, 2, or 3. There were questions about the past and future, and I'm going to show you those in yellow in the next slide. In contrast, the theory of mind task, they had to read short stories and then answer questions about them. For example, when Christine labeled the pie at the school's bake sale, she thought it was cherry and labeled the pie cherry. Actually, the pie was strawberry. A customer purchasing the pie expects it to taste like cherries. True or false? So to do this task, the participant has to put themselves in the mind of the customer in this case and think, what did they know? What would they be thinking? So this is why it's called theory of mind. And I'm going to show you activations from those tasks in red. The question is, you can already see here in this map that they seem to occupy different regions within these individuals. But the question is, how do these relate to the default network A and B that I've been talking about? And so here is the results from one individual, subject 7. Network A and B are shown here in light blue and dark blue. And now I'm going to take the boundaries from network A, which is the dark blue in the top image, and I'm going to show them in black here on the left. So these black boundaries here are from network A, and you can hopefully already see that there is a large amount of overlap with these yellow regions, which represent activation for the episodic task, so thinking of the future or the past. So network A seems to activate, seems to overlap with the yellow regions for the episodic projection task. And you can see that in some cases, it's a very close overlap. Like the boundaries of the yellow regions are exactly where the black boundaries from the connectivity derived network are. If I then take the regions of network B and show you them on the right, you can see that actually those are overlapping with the red regions that are activated for the theory of mind task. So you have this functional dissociation already that network A that's defined at rest using connectivity is showing quite specific activations for this episodic projection task, whereas network B is showing activations for this theory of mind task. And one important thing that I wanted to talk about is that this overlap seems to be throughout the entire network. And this is an important point. It's not the case that just one region of network A is showing activation for the episodic projection task. Actually, it seems as if the distributed network as a whole is specialized for episodic projection. And this is just to say that we replicated this within 18 individuals, I think. It wasn't perfect, but across individuals, there definitely was evidence that each part of the network is activated for this task, for these 2 tasks. And so where that ties in with the story is that we have evidence that these 2 networks are parallel and distributed, but now we also have evidence that they are specialized for different things. And maybe that's why they are separate networks and how they came to be organized this way. So I've just presented evidence for functional specialization of parallel networks. And in this context is where I became interested in studying how do language regions fit with this organization that we were seeing. And so I then turned to studying within these same individuals what the language network might look like. So when I was searching for these networks in the data set I just showed you, I often observed this network that I'm showing you here. So the seed I picked is right here in the middle, posterior middle frontal gyrus. And you can see this correlated network that seems to just resembles a language network. It has Broca's area in the IFG, it has Wernicke's area here, it has some evidence of regions in the temporal pole, and also this region in SMA, which I wasn't aware of at first, but apparently that speech SMA it's a region that's known to be part of the language network. And so we decided to explore this further. Here is another individual where I'm picking the seed, and it's showing a map that's a little bit more noisy, but it still covers the same set of regions. Broca's, Wernicke's, temporal pole, and SMA. These are seed-based correlation maps. We then confirmed that we could see these just using a simple clustering approach. This is k-means clustering. And again, you can see, I'm going to just highlight those, Broca's, Wernicke's, temporal pole, and speech SMA in both of these individuals. But what was interesting is that there are also these additional regions. You can see them already here, one in the posterior cingulate cortex, inferior temporal cortex, and even this tiny region in the ventromedial prefrontal cortex that we didn't expect to be part of the language system, but there they are. And so where this becomes interesting is if you compare the organization of this network, this candidate language network, with the organization of the networks I've been talking about, the default network, these also seem to occupy multiple regions of the brain in this distributed fashion. And in each of these zones, you can see that the language network sits beside the default network, closer to default network B in this case. Not only that, but you can often see these sequences from yellow to pink to red, yellow to pink to red, yellow to pink to red, that occur in multiple parts of the brain. So it seems that this candidate language network is also another example of this parallel distributed network that just occupies slightly different regions. So I've just shown you evidence that the language network is a typical association network. The question is then, is this actually a language network? Is it actually activated for a language task? And so to test this, we did a clustering analysis where we clustered the brains of these individuals into 17 networks, and we picked 6 networks a priori to test. And these were networks that were selected based on having been implicated in language in some form or another in the literature. So we had the default networks in there, we had the frontal parietal control networks, and we had the salience network. And these are all networks that sit really close together to the language network. As you can see, throughout most of the brain, these are really, really densely packed networks, but defined by connectivity, they're separable. We then had these same individuals undertake Ev Fedorenko's language localizer task. And in this task, participants stare at the screen, and then they are presented with sentences, one word at a time. So the sentence might be, a rusty lock was found in the drawer. And at the end of each sentence, they just had to press a button to say that they were paying attention. So that's a sentence. The control condition is this pronounceable non-words condition where it's the same thing. They're just shown one word at a time, but these are, in this case, it doesn't form a sentence. And then they have to press a button at the end of the non-word list. So here is the task activation map for this contrast for this same individual. And when I saw this, I was really surprised at just how closely the task activations from a different data set overlap with the functional connectivity maps from this same person. So you can see the boundaries actually overlap really closely. And almost the whole of the network seems to be showing increased activity for this language task. It's not perfect. There are definitely regions outside it, but these seem to be less consistent across individuals when we compared across individuals. We then took the average signal for each of these networks and plotted it. So to show that indeed the language network that we selected a priori seems to have much higher activation for this language task. And that was consistent across, we did 18 individuals in total. But in each case, you can see, the overlap is really close between the task activations and the connectivity. Another feature that I commented on earlier is that throughout the network, so even in these smaller regions that are not part of the classical language system, you see evidence of task activation. For example, here in the dorsal posterior cingulate cortex, or this ventral medial prefrontal cortex. Especially when we looked across individuals, each of those smaller regions showed some evidence of engagement during the language task, which was unexpected. So it seems as if this language system as a distributed network is also specialized as a whole system. The level of specialization is at the level of the network, which is was very exciting. And just a couple of other things I wanted to leave you with. One is this idea that, I'm arguing that the language network shares the same broad arrangement as the other association networks, but there are clear differences. For example, here in the posterior medial cortex, the default network regions are very large, right? This is in inferior portions of in the retrosplenial cortex, these receive a lot of projections from the medial temporal lobe. And so if default network A, shown in red here, has a role in memory and in autobiographical episodic processes, then is that why the region of the default network here is so large? In contrast, the language network has a very small, diminished region in this part. But if you look in the lateral temporal cortex, this region is really large for the language network, and also in inferior frontal gyrus, the language network has a very large region. Is the size of the regions within this distributed organization, does that tell us something about its function? And this is not a new idea. Here's work by Leah Krubitzer, where she showed how within different species, how behaviorally relevant that sensory organ is, changes the size of the cortex that's devoted to processing that sensory modality. So in the platypus, there's a large portion of its brain devoted to mechanosensory and electrosensory information. So, the cortex is growing because of the relevance of that sensory modality. And in the humans, it's been argued that Broca's area sits where it does because it's so close to regions of the somatomotor strip that control orofacial muscles for articulation that are important for language. So we decided to explore the organization of these regions within these individuals. Here we have the language network for subject 1. And in this subject, we also had them doing a motor localizer task where they were moving their fingers, where they moved their feet, but also moved their tongue in their mouths in a way that minimized their head motion. And so here is the task activation map for them moving their tongues in the scanner compared to moving their fingers and feet. And you can see that, as expected, it covers the inferior part of the somatomotor strip. And it's bilateral, which is important. But there is this interesting gap between the language network and this tongue motor region. And so we wanted to explore what was going on there with functional connectivity. So I'm going to switch to a functional connectivity map now. I'm going to place a seed here in the contralateral hemisphere so that you can see the correlations in the left hemisphere. And you can see, just if I flick back and forth there's a good bit of overlap between the high correlation regions and the task activation regions. But yeah, so here is that motor map. And then we have this gap where I placed the seed just to see what was going on. And this is the network that we saw, that it seemed to bridge the language region that was closest to the tongue motor region, but it also showed connectivity to these other small regions that were bordering the language network in multiple parts of the brain. So here in SMA, here in the Wernicke's area, and this other region in IFG. And then if you look at these networks in sequence, they seem to form a sequence that links tongue motor regions to the language network. Now, I can also go over to auditory cortex and show that this intermediate network also bridges the language network and auditory cortex, this time defined with a seed in the contralateral hemisphere. So why this is interesting is because the language network seems to be part of this hierarchy of networks that bridges both auditory and orofacial somatomotor regions, which are important for language. So to me, it suggests that that's why the language network sits in this part of the brain, because it is so close to these functionally relevant systems. So yeah, and to summarize that, I've just shown you evidence that local functional regions might influence the specialization of the distributed association networks. And that basically ties in with this idea that association cortices, the networks that sit in association cortex, they inherit this broad organizational motif, but then they're specialized by activity-dependent processes. So to conclude, I have showed you that individual-focused analyses in fMRI can reveal new details of brain organization. I've shown you that a language network can be reliably defined within individuals using functional connectivity. I've shown you that the language network is a typical association network in that it shares a common distributed motif that interconnects multiple association zones. And I've shown you that this entire language network shows evidence of increased activity during a task broadly targeting linguistic processes. And finally, I've shown you evidence that the language network is part of a hierarchy of distributed networks that link language, orofacial, somatomotor, and auditory regions of the brain. And I'd just like to thank the many people that helped me with this work and made this possible. And thank you for your time.
All right. And Dr. Kahan, I think you are going to help facilitate. I don't see any questions submitted in the chat right now.
Yeah, let me start with a question, if I may. So, Brad, I think the ability to do single subject studies with functional imaging, that's extremely important for obvious reasons that you outlined. But like everything else, good things come with compromises, that sometimes it is good to average across because there are individual idiosyncrasies. You know, when we give somebody a task, we assume that the task is the same for all, but it doesn't have to be. Subject A may take another strategy than subject B. And then when you see a certain distribution, you can't really conclude that that's the way that the subject is performing the task. So the question is, how do you deal with individual differences? And is the method, the technology you're describing with this beautiful SNR, is that a way to finally start to study the neurobiology of talent? You know, there are people who can sing, there are people who can run, there are people who can taste wine, and these are different talents. So how do we approach that? How do we approach not just the difference in the folding of sulci and gyri, but also in the remarkable individual differences that separate one person from another?
Yeah, it's a really great question. There are a couple of things to say. One is that in order to understand the individual differences, we have to first be able to get a clear map of the anatomy within that individual. So, you know, this is a first stab at understanding these networks, at least with this within individuals. But I think if we had, say, 1,000 people with, you know, this level of mapping on them, maybe we could then start to look at individual differences, because you're going to need larger cohorts to do those kinds of analyses. But I absolutely agree that that's a very important point. I think this is just the first step towards getting at that question, by being able to look at the organization within an individual. I am very curious to know whether this idea of the, you know, one region of the network expanding, whether that has any behavioral relevance. It's possible that that's more of a, you know, that it's still a very blunt look at the relationship between the activity and function, because maybe this is determined during development, and it doesn't really, you know, if you can, if you train on the piano, you're not going to see a growth in the size of these regions. Maybe these are a lot more stable than that. But I think it's an empirical question. It's something I'm definitely curious to know, whether musicians just have, or, you know, journalists have larger language networks than others. But another thing to say is that I think, you know, this task is very broad. It's just reading words with meaning versus reading words without meaning. It doesn't get at a lot of the things that linguists are more curious about, which is, you know, differences in grammar and articulation. It's just broad. Am I looking at language? Am I processing language versus am I just looking at squiggles? So I think we need to tone down our expectations of the data in that sense, at least until we get better methods.
Well, let me then ask another question, quite different.
Mm-hmm.
So you showed us in your language network the left side. What did you see on the right?
Yeah, I wish I had put some slides in, but we saw that in every case, it was a bilateral network. So
well, okay, so then let me follow up on that. But in the human, you make huge lesions on the right, language remains intact.
Right.
So what do those activations on the right mean?
Well, so again, I'm working on the hypothesis that the language network formed inheriting this broad connectivity from association cortex. That's just a typical prototype. And then during its specialization, the left hemisphere regions grew and the right hemisphere regions shrunk. That's one thing I didn't say. The right hemisphere regions were all a lot smaller than the left ones. And so maybe those are working together. And if you have a network region and you chop a little bit out of it, maybe the region can still function. But if you take the whole thing out, it's more problematic. And maybe that's something that we're seeing in right hemisphere lesions, where you can take out the right hemisphere, but you still have a lot of the homologous region on the other hemisphere, which can do its function. It's just one idea. I'm really not sure. It's very interesting to explore that.
Any other questions? Well, I have one actually. We're kind of alluding to this earlier, but do you suspect that, say, these parallel networks, that they maybe were originally one, or maybe continue to be one discrete network, but just later developed functional parcellation? Or do you think that the functional parcellation between these parallel networks was kind of baked in, during neural development?
So yeah, this is an idea because if you look across individuals, there's a lot of variability in the shape, in the exact location, even in the number of small regions. Some people will have multiple regions and others will have 2 or 1. So what kind of processes would lead to this complicated features? And the idea there is broadly, this region is going to specialize into language given its nearby inputs from, say, auditory or tongue somatomotor regions. But actually, which exact neurons become specialized or how those regions group together maybe is an activity-dependent process, which leads to this complicated structure. So, I think maybe the answer is both. Yes, in some ways, it's predetermined that these are going to specialize, but the actual, detailed topography is, I think, less specified.
All right, any other questions?
I had a quick question for you. Thanks for the talk, Rod. So, just as you're showing that, group averaging is smoothing spatially, fMRI is smoothing temporally. So how much of these generalizations that you're making about networks do you think is affected by the poor temporal resolution of fMRI versus, especially resting state, versus something like ECOG or stereo EEG? So I mean, I guess I'm getting at how much do you think these generalizations will hold when we get better temporal resolution?
Yeah, it's a really good question. I do think that even within the slow resolution of fMRI, we are filtering the data to be to look at slow oscillations, and that's what we're correlating. I think by doing that, we are specifically testing more stable features of connections. I think that's why we see what I think is the closest relationship to the projection patterns that we, from the animal work. I think that that's the basis at which higher frequency things are occurring. So if you know, and you can even see with ECOG, if you do a slow filtering, you can see high frequency slow oscillations that seem to match the bold oscillations as well. But that's just one part of what's going on. If you can then, within these, within this organization, you can have faster oscillations and faster interactions, which I think is super important. It's just another question in a way. I don't think that's going to deny what this is showing, right? It's just a different level of description.
All right, anybody else? Well, I guess with that, well, thank you, Dr. Braga, for your wonderful talk. Really appreciated hearing all that, and have a lot to think about following this lecture. And we look forward to working with you in the future.
Me too. Thank you so much.
All right, take care.